Okay. Now, there's also something called non-random mating. Oftentimes it's like mating with like. Sometimes it's like mating with different. So if you go to certain places, let's say Iceland. Iceland as a whole is it's a isolated population. They have alleles that are probably not found anywhere else. A lot of Icelanders look at the same, they're very similar. They are mating like versus like just by chance. Maybe if there was other choices, they would. Or you could have a situation where, you like a certain kind of partner. So you can have assortative mating, or you can go the other way around, let's say. You can have it doesn't show disassortative mating, where actually you might get different colors. So you might go this, and that one goes with a red one, okay, instead of a blue one. Okay. Now, or you can have random mating. If you have random mating, well, when you have non-random mating, you also tend to increase drift because you have more individuals that are alike because they share the same alleles from the same ancestor and not. Okay, now, one of the problems with assortative mating is you end up with something called inbreeding depression. So, good examples of that are matings between close relatives. In other words, because they're related, they share alleles because they have a common ancestor. So, obviously, if you have a brother and a sister mating, they're going to share a lot of alleles because they both have the same mother and father. You can also have outbreeding depression. That would probably happen because of disassortative mating, perhaps migrants coming in, and that could either bring new alleles in and new allele combinations. That might be good, or it might lead to outbreeding depression. In other words, the two populations, you have dispersal from one into another, perhaps as a plant, and phenology differences. So when the plant actually blooms, in one it's a high mountain late summer bloom, the other is low in the valley and let's say early summer or midsummer bloom. Egyptian mummies and the royal family. Royal matings were tightly controlled. They were often a brother and a sister mating to produce offspring. Think about what happened on that island. That was non-random mating. So what often happens is you start seeing a lot of recessive genetic problems in individuals where there's inbreeding depression because there's mating similar to each other, they're related. In fact, they're sharing the same alleles that they got from, let's say, their grandfathers and their great-grandfathers and all that. And there's a chance that they're also sharing the same mutations, and if they're recessive mutations that cause a problem, then they show up. So, in terms of King Tut, his parents were brother and sister. He, from looking at his mummy. He had a club foot, necrotic foot bones. He had a cleft palate. He also had weaker immunity, because oftentimes with immune systems you like as many different kinds of alleles as possible for recognizing different infections. And then he also was the father of at least 2 stillborn fetuses. In other words, he was a wreck. He died at 19. He might have tried to mate with someone. Probably not someone that you would choose, because he was a wreck, but of course he was the son of God or whatever. So, you know, maybe it would have been good. Now, the same thing happened in royals in Europe. All those European families, even though they went to war together, they all were related. And you tended, in a lot of royal families, you saw the same sorts of things. There were some sex-linked traits where a greater incidence of color blindness, a greater incidence of hemophilia. These are all on the X chromosome. So it was mainly the boys that showed it, because the boy only has an X and a Y. If you have 2 X's and there's a mutation, you get a rescue effect oftentimes on the other X. So it might be one bad allele, but the other allele makes up for it. But if you have an X and a Y, there is no rescue effect. That's why males in humans and in a lot of mammals show, or tend to show genetic problems before females do. So, all you have to do is read European history. A lot of kings and queens were obviously, they were crazy. All kinds of mental health issues there too. Okay, and then there's also another thing that changes allele frequencies: migration. One population, individuals migrate from one population to another. So you can bring in new alleles. New alleles can leave from the population, one population, and go somewhere else. Gene flow also includes dispersal for organisms that can't move around on their own. Perhaps, for example, we have pollen from plants, and either the pollen blows in the wind to another plant, or an animal comes along, if there's showy flowers, and gets pollen on it and goes to the next plant. This is coral dispersing either eggs or sperm, and which one will do eggs and one will do sperm, and there'll be fertilization in water column, and then they'll float away. There's also seed dispersal, like this and like this. A lot of times you can actually have seed dispersal where animals carry it away. In this case, we have wind dispersal here and water dispersal here, but the most common is seeds that are dispersed, or they eat the fruit and they ingest the seeds, and then as they move, they disperse it all over the place. So, and it causes what's called gene flow. You have population 1 and population 2, and now if they start dispersing back and forth, you end up with population 2. Both of them start looking more and more like each other. They've got both alleles. So when there's gene flow from migration, populations that may be different start to look very, very more and more similar in terms of allele frequencies. When gene flow, this is important, when gene flow is interrupted, that's when populations become isolated. So they may have been one big population, something stops gene flow, and each population starts to evolve separately, whether it's because of a different environment or it's just because of all these other forces of evolution, like genetic drift. So this is the Grand Canyon. The Kaibab squirrel. The thing is, the Grand Canyon was split starting about 70 million years ago. And there used to be just a continuous plateau. Okay, there was no big canyon. So the population of squirrels were all the same, identical. Well, eventually, those on the south rim, there was no more dispersal or gene flow going to the north rim, and they evolved in isolation. And now there's two different species. We have the species with these funny ears. Can't really see, but on the north rim. And then you have these that don't have such funny ears. They have now become actual different species. So, in terms of the take-home, is that if you stop gene flow, you end up with what's called reproductive isolation. That means you have two populations now that has been split, and each one is likely to diverge just by chance alone, just from genetic drift and things like that. Or they might be split and you end up with different environments too, so selection is also happening to the two different populations. And eventually they become separate species. I do want to say one thing initially. I hope I'm not overriding this. We're looking at a population here. In this case, a population, let's say, of dogs or anything like that. And a population has what's called a gene pool. And that means all the alleles in the gene pool in the total population. So you have these dogs, and remember they have two alleles for each locus. So they may have two of the same alleles. Those are homologous alleles. Or they may have two different alleles. And so if you mix them all in, and there's random mating, you're dealing with a gene pool. And gene pools affect how things happen, or the strength of certain things like genetic drift, or if evolution by natural selection becomes efficient, things like that. And remember, you've got small, you're likely to have more genetic drift, and you're likely to have more problems with genetic drift. So, in terms of a total gene pool, if there's a new selective pressure, and it's a small population, there's a greater chance it'll go extinct. But if there's a large gene pool, it will survive, but it will change genetically under, let's say, selection. So, a species with a large gene pool, if you have a large gene pool, natural selection can work much better in a large gene pool than a small gene pool. In a small gene pool, drift is so strong, genetic drift, that even if there's strong selection, just by random chance alone, a good allele might actually disappear. So gene pool is one of the things you have to understand in terms of how things change over time. The bottom line is The gene pool is essentially a population of alleles, of genes. Since diploid organisms have 2 alleles for every locus, one from mama, one from papa, there'll be double the number of alleles in the gene pool than compared to the actual number of individuals in the population. The thing to remember is that in large gene pools, like large populations, drift is much weaker. And actually, evolution by natural selection is much more efficient. Instead of losing important alleles, like you would in a small gene pool from drift, selection can act on those alleles that affect fitness and either weed out the ones that aren't as adaptive as others or favor the ones that are adaptive. Now, in a small gene pool, because drift is so strong, drift can actually override selection. So you may have an allele, a mutation, an allele, a new allele that actually increases an individual's fitness by a lot. But if you only have 10 individuals and there's only one allele out of 20 in the gene pool, that allele, because drift is so strong, it might go to fixation, or it actually might go extinct, regardless of how well selection acts or how strongly selection acts on it. So in the face of environmental change, a new selective pressure, which is more likely to go extinct? A species with a large gene pool or a species with a small gene pool? Well, as I mentioned, selection works in large gene pools. Populations can evolve. Small gene pools, there's fewer alleles, usually, and the chances of a losing allele or one allele going to fixation are so great because of drift that the species with a small gene pool is under much greater probability of actually going extinct. Okay. Darwin's second great idea. Descent from a common ancestor. Do you have any brothers or sisters?
Yeah.
Do you look like any of them?
Yeah.
Okay. Why? Why do you look more like one of them than like him over there?
Because we inherited traits from the same person.
Okay. So, different parents, descended from different ancestors. Okay. So that's why family members generally look more like each other. Not all the time. Sometimes there's been a little dilly-dallying on the side, and all of a sudden there's a nice blonde-haired, blue-eyed kid that shows up, and there's never been that in the family. But, okay. Now, the same thing can be said about species. They also are descended from the same common ancestor. That's why certain species resemble each other more than others. Think about the dog family. Okay, there's dogs that we've domesticated, but there's also wolves, gray wolves, a whole bunch of different kinds of foxes, wild dogs in Africa, a whole bunch of different species. They all look similar to each other in certain regard, but not identical. But they certainly don't look like a cat, and they really certainly don't look like a bacteria. But they actually share a lot of stuff with a bacteria. They use the same exact genetic code. So all life that we've found on Earth at this point is descended from a common ancestor. So when someone says, oh, I didn't descend from a monkey. No, you descended from bacteria. Okay? Okay, so why might descendants of either resemble their ancestors? They share the same genetics. Why are neither descendants of families or species identical? Well, all the forces of evolution we've been talking about. Genetic drift, natural selection. But something has to stop gene flow between one population and another, or a large population has to be split for that to happen. So oftentimes that's why you got two different species of squirrels, one on the north rim, one on the south rim. Starting around 7 million years ago, dispersal and gene flow between the two became less and less and less common until it was finished. Evidence for common ancestry. So this is one of Darwin and Wallace's second great idea, we'll say. The first was evolution by natural selection. The second is that all life, it turns out, is actually related to each other. And we know this for a number of reasons. Now, you already had some of this when you actually looked at that video of Jerry Coyne talking about evidence that evolution is true when we first started on evolution. But the first, we'll say the first lines of biological evidence point to common ancestry. Origination and extinction in a fossil record. So if you see some a taxa, and then all of a sudden it's not there anymore, or a new taxa originates at a certain point in the fossil record. And remember, the fossil record is a record of deposition of fossils over time, from very old to much more recent. So when you have origination and extinction in the fossil record, oftentimes you'll have transitional taxa. In other words, taxa that show transitional forms from one form to another. For example, birds are actually dinosaurs, and there were actually bird-like dinosaurs before there were birds. And they had feathers, among other things, but they couldn't fly. And so, like, Archaeopteryx is a good example of perhaps the first bird, but it still has traits that are, we'll call primitive, and it has traits that are new. So primitive traits are ancestral. In the case of Archaeopteryx, well, they have a tail. Birds don't have a tail anymore. Tails were not favored in the bird lineage for flying, and they disappeared. Birds also have teeth, or Archaeopteryx has teeth, but birds don't have teeth anymore. They have bills. Well, teeth are heavy, and so they were not favored as well for a flying animal. So we have Archaeopteryx. It looks like it was able to fly. It has feathers. It has the kinds of feathers that you normally see, flight feathers that are asymmetrical and affect lift. But at the same time, you have, and those are new features. And they have feathers, right, and different kinds of feathers, flight feathers. So Archaeopteryx is a mix of ancestral traits and new traits. Okay, then secondly, homology. This is any similarity between species or other taxa, whether it's families like dogs, the dog family, or the cat family, Felidae versus Canidae, any similarities that are due to evolution from common ancestry. In other words, the taxa we're looking at had a common ancestor, and there's certain traits that they share that other groups do not share. So for example, if you have land animals and land vertebrates. Land vertebrates, they have 4 legs. We have 4 legs or 4 arms, 4 limbs, not whatever they are. They're different for all these different taxa because they're used for different things, but we actually, all vertebrates on land, amphibians, different forms of reptiles like lizards and snakes, turtles, crocodilians, dinosaurs, birds, all have 4 limbs. So that shows that the most parsimonious, simplest explanation is that there was a taxa that at one time emerged from the water, like a fish, and there was selection so that some individuals, let's say living in an estuary, if they could move around a little bit, they probably had a better advantage. And so eventually you had these transitional forms where you finally ended up with the first land animal with 4 limbs. So homology, anatomical similarities, 4 limbs in terms of all vertebrates on land. Vestigial traits in modern species. In other words, vestigial traits no longer serve their original function and oftentimes are just very small compared to what they used to be. So for example, whales, or actually, we'll look at this in a second, but whales are actually more related to hoofed animals than anything else. In fact, and we know this because if you look at the skeleton of a lot of whales, you'll see the remains of limbs. And for the hind limbs, like the pelvic girdle, things like that. Similarly, shared developmental pathways. As an embryo develops, it often develops in a way that kind of redoes or copies what happened during the evolution of the species. So with humans, we start out a bunch of cells, they start developing, changing, you start differentiating cells, and it turns out we're vertebrates. We share an ancestry with vertebrates going way back in time, all the way beyond the earliest fish. And we also, of course, shared similarities with fish, which are all related to us and every other vertebrate. A lot of fish have gills. When humans actually develop at a certain point in the development of the fetus, you can actually see gills being formed. They don't do anything, but they're formed. And then as the embryo develops more and more, the gills then disappear and lungs take over. The same thing with tail. We don't have a tail anymore, but the embryo at one point has a tail. As Coyne showed, our most recent common ancestor was covered in hair. Well, babies at a certain point, or fetuses, they actually get covered in hair and then it disappears. So this is shared development. And it's all because we share the same ancestors. And then there's DNA similarities for the same reason. You, just like you and your, let's say, siblings, if you have any, you share the same DNA because you got it both from both your parents. You share parents. And so there's DNA similarities due to ancestry. And then finally, there's distribution of organisms on Earth. It's called biogeography. And one thing that Darwin noted and Wallace noted were that in certain areas, even if you had organisms that didn't necessarily look all that similar, they often were. So in Australia, why is it that almost all the mammals in Australia are marsupials, like the kangaroo? Now in North America, we have a marsupial. It's called an opossum. It has a pouch. That's the only one. In South America, there's a number more, diversity in terms of marsupials. But the greatest diversity is in Australia. This is all because South America, Africa, Australia, all formed this large continent called Gondwanaland. Now, probably the first mammals that showed up were egg-laying mammals. Then, over time, the marsupials showed up, and it looks like perhaps the placental mammals, like we are, live births, no pouch, all that. It looks like they evolved later. Well, remember, Gondwanaland was a giant continent, but it broke up into smaller continents. It broke up, part of it was Africa, part of it was India, part of it was South America, part of it was actually Antarctica. There's fossils there. Part of it, and then part of it split up and became Australia. Well, when they split up, it was different for each different part. So depending on how far or what kind of mammals they had at that time, what had evolved, that's what you generally see. Well, Australia split out the quickest, and essentially before humans arrived, there were really no other mammals except marsupials and a few egg-laying mammals. Placentals, by the time Australia split off from Gondwanaland, placental mammals hadn't evolved yet. There had only been egg-laying mammals and marsupials. So we'll look at that biogeography as well. One evidence for common ancestry is looking at geological time. Which organisms started to appear first? Which organisms appeared later? More recent organisms. So, way back around 3.8 billion years ago, the first oceans the planet was still being bombarded with asteroids and all sorts of different things. Not much was going on. There was some liquid water on Earth. Around 3.5 billion years ago, we actually have fossils that look like a certain kind of bacteria, and it looks like the origin of life. Or in other words, the origin of life had actually started probably prior to when we actually saw it. Then we start seeing things like the evolution of photosynthesis. In other words, animals that could turn the energy in the sun, or not animals, organisms that could turn the energy of the sun into carbohydrates. And those first were certain kinds of bacteria, and then much later plants. First, cyanobacteria. We still have these around today, and they are bacteria that are actually they're using photosynthesis to produce fresh compounds. And then other things that don't produce their own actually probably eat them. Now, one of the byproducts of photosynthesis is oxygen. And so over time, over millions and millions and millions of years, the first evidence of atmospheric oxygen starts to show up. Then there were also the first eukaryotic cells. Eukaryotic cell, bacteria and archaea, they don't have a nucleus and they don't have organelles. But eukaryotic cells do. They have a nucleus and they have organelles like mitochondria and like chloroplasts for photosynthetic cells. In fact, it looks like eukaryotic cells began when different life forms actually started living in a symbiotic environment. So it turns out mitochondria that we have and that other multicellular organisms or eukaryotes, some are single-celled, have, we found out that they are most common to a certain kind of bacteria. It looks like the original eukaryote somehow acquired mitochondria by engulfing or being invaded by another bacteria. The eukaryotes that can actually make oxygen, photosynthetic, looks like they, as well, got their photosynthetic bacteria. They're actually related to cyanobacteria. Same sort of thing. And then the nucleus, there's a bunch of different hypotheses how a nucleus actually arose. The most common one is it was a virus. Okay. So then we had first photosynthetic eukaryotes. First evidence of sexual structures in red algae. These are multicellular eukaryotes. There's still a lot of single-cell eukaryotes running around. And then right around this Precambrian, up until about 550 million years ago, you finally start to see the first animals. And also the first lichen, which are a symbiosis between plants and fungi. Also, the amount of oxygen went up, and even the ocean was oxygenated. And so the first signs of life are these little sponges that they found. Actually, they found them all the way back almost 750 million years ago. Then there was an explosion of life of multicellular animals. And if we actually look at this, all these different kinds of taxa, including jellyfish, including other sponges, including cnidarians, sea anemones, mollusks, all the major taxa, arthropods, including spiders and insects, and also the first crustaceans. Crustaceans including, well, actually, crustaceans came before insects and stuff like that, found in the ocean. Think about lobsters and things like that. And also the first vertebrates started showing up. Okay. Now, the first land plants show up just after the Cambrian, and the Cambrian was called an explosion because all of a sudden there was an explosion of multicellular animals. So the first land plants started showing up between the Cambrian and Ordovician, and you can see this in the geologic layers of rock. First land plants. The Silurian, the first bony fish. And then the Devonian. We have a lot of things happening during the Devonian. First insects, first fish with jaws, first ferns, vascular plants, first tree-sized plants, first winged insects, first tetrapods, amphibians, in other words, vertebrates that have 4 legs. In other words, the land had finally been invaded by the vertebrates. First seed plants, in other words, plants that actually made seeds instead of using spores, and they actually had leaves to use for photosynthesis. During the Mississippian, in the Carboniferous, the first reptiles have evolved. And these reptiles, they were actually the first that had an amniotic sac around the egg. Then, at this area right here, this is the first mammal-like reptiles start to appear. In other words, it looks like, well, they probably had some sort of fur, and of course, mammals also mammary glands and milk, so there was something like that. Then plants started evolving vessels. These are all traits that start showing up. Now, there was also these big extinctions that we start seeing. Ordovician extinction between the Ordovician and the Silurian. That means a lot of animals that were alive during the Ordovician period went extinct. And the Silurian was basically made up of animals that had actually survived and then speciated. Similarly, the Devonian extinction, same sort of thing. A lot of taxa went extinct, and the ones that actually didn't then gave rise to a bunch of new taxa. So the first reptiles, the first mammals. The biggest extinction was the Permian extinction. Over 90% of all species in the ocean went extinct. It was huge. Started around 250 million years ago. Now, in the Triassic period, we have the first nectar-drinking insects. In other words, we have plants that are using insects to pollinate. We have the first dinosaurs. The Jurassic, the first real mammals, probably egg layers. Tyrannosaurus rex. The end of the Jurassic. We have there was also the Triassic extinction. So between the Triassic and Jurassic, there was a huge extinction event there. And then each little era, like we have Jurassic, Triassic, there was always a turnover in the kinds of organisms. And so each of them actually probably had a certain amount of extinctions going on that differentiated. But the really big ones, we have them laid out. There's 5 of them. There's actually, before the Cambrian, there were probably a lot of big extinctions. Let's see. So first, angiosperms, flowering plants. First, diatoms, water lilies. First, magnolia family of plants. First, bee. First, ant. First, placental mammals. And of course, during the Cretaceous, there was lots of dinosaurs and various things. And then the Cretaceous extinction happened. It was not one of the biggest, but it was big, and it looks like it was caused by an asteroid striking the Earth. And it wiped out not only the dinosaurs, it wiped out a whole bunch of other things. The only dinosaurs it didn't wipe out were birds. Okay. And then, more recently, during the Cenozoic era, all the way up to our modern era, it started out with the Paleocene, the Paleogene, were different things. First fully aquatic whales, first horses, first primates, first rabbits and hares, oldest pollen from daisy family plants, Oligocene and the first apes, first orchids, earliest homonyms, in other words, besides a split from the apes that gave rise to all sorts of human-like organisms. And then at the Holocene, at the very latest, of course, Homo sapiens have been around, probably only a couple of 100,000 years. So we can always, if you remember that movie, we can always predict in the fossil record where we'll find certain types of fossils. We will not find rabbits in the Cambrian 550 million years ago. If we did, the theory of evolution would be wrong. You would be able to refute the theory. It's never happened. So, if we look at the earliest, here's millions of years along the bottom. From more recent to earlier, older. The first jawless vertebrates, in other words, they were vertebrates, but instead of having jaws, they didn't. They had other ways of feeding. Showed up right around here, around the Cambrian. There were some extinct jawed fish that went extinct. So the first jawed fish. Later on, you can see where they originated. Sharks and rays. Then the bony fish evolved much later. And then finally, some of these bony fish, or some fish, actually evolved into tetrapods, land vertebrates with 4 limbs. And you can see that right around here. And then the reptiles started showing up. The mammals split off from the reptiles earlier than the birds did. And birds were actually part of other taxa, including they were part of dinosaurs. And they were also much more closely related to crocodilians as well. So you can see how certain things show up in the fossil record at certain times. And we would not, that's, when you think about it, like creationists, they say, human beings and dinosaurs lived at the same time. Well, they didn't. And there's no evidence of that whatsoever. There's no fossil evidence. There are no imprinting evidence, nothing like that. So, transitional taxa. These are kind of intermediate organisms between one form and another. So the first we'll look at is fish, and the evolution from fish to the origin of tetrapods, vertebrates with 4 limbs on the land. In the beginning, we had what are called these lobe-fin fish, right, like here. And they had fish-like traits for life in the water, but they also had things that would later be used for actual limbs, because they're homologous structures. A lot of these fish lived in very shallow waters, and then over time, we finally have kind of a tetrapod that started looking more like a fish, but still now we've finally got some limbs. And not only do we have limbs, but and pelvises and all that. Here's a remnant of a tail. This animal was clearly living in water and probably coming out onto land. This animal also had a neck. Fish don't have necks. They can't turn their head around. This guy probably could. So this was the first early tetrapod species. And then finally we started getting into Tiktaalik. And this is clearly has fish qualities and tetrapod qualities. Let's see. The gills are still present. 4 limbs are fins with webbing and no external digits. Okay? So they could stand on them, and they probably use them for swimming in really shallow water. The pectoral fins are weight-bearing, with distinct jointed wrist and finger bones, right here. Head is long and flat, instead of like a fish. The neck is mobile, not fused to the shoulder. The ribs are long and sturdy to support the body out on the land. Small pelvic fins are present. In other words, they'll be the back legs. A tail, kind of like a fish tail. Scales are present. So this is the evolution. These are intermediate taxa that we found in the fossil record, just where they should be, actually. And then finally, we have intermediate fossil in terms of birds, the evolution of birds. In fact, okay, so this is Dromaeosaur. It actually supports, it's actually a dinosaur. It's got feathers on it. It's got sharp claws, a long reptilian tail, lots of feathers. A lot of them also appear to have feathers that are of different colors. So obviously it wasn't flying yet. It had feathers mainly for downy feathers for thermoregulation. They might have been warm-blooded, or and they also might have used their feathers like birds do today for display. And then finally we have Archaeopteryx. Still got the tail. That's an old trait. Still actually have teeth instead of a bill. But certainly we actually have now flight feathers. And flight feathers are different than other feathers in that they're asymmetrical, so they can actually create lift during flight. And we've also talked about, this is a phylogeny. This is actually shows evolutionary relationships. And we didn't know where to put whales before, because these phylogenies were based on physical features. But with DNA, we've now found out, in fact, that whales don't have an astragalus, which is part of their ankle. They don't have an ankle. They don't have an arm or a leg. So if you use astragalus, either having it or not, as a trait that you could group things together, it would be misleading. But we have a whole bunch of DNA that's identical. And so we can actually show that whales are part of these artiodactyls. In fact, it turns out probably the closest living relative to a whale now is a hippopotamus. Here I'm going to introduce you to phylogenies. So phylogenies are a history of evolutionary change in various lineages. It goes from old to new. So, and every point like this where something splits off means that now there were two different species. There was species A and species B, and there was some trait that A had, whereas the remaining clade, the group, there was just one species. We see three, but it was actually just one individual species back then, and then split up. So we have a most recent common ancestor for A and this whole clade. So at this point, let's say we have, let's see, fish. We'll look at tetrapods like we did already. We might have a fish here. No limbs, vertebrate, fish-like tail, fins, all that kind of stuff. The same thing, another most recent common ancestor for for B. So now there's this lineage that was just one species that split into two, most recent common ancestor, and we'll say that the trait that defines it, actually forget about that. Okay, so there's fish. We might have here the beginning of limbs. And then up here, we might have the beginning of, let's see, what kind of limbs? Oh, we'll say scales and reptiles. Okay, so actually amniotes. So the reproductive embryos are in a sac, and these animals can actually live far away from water. And then finally, we have another up here, and so we have reptiles. We might also have, sorry. Okay. And then we might have something that reveals a split, and perhaps these are feathers. And that includes, and a bunch of other things like a four-chambered heart. So that's going to include not only birds, but dinosaurs, certain kinds of dinosaurs. And then here, for mammary glands. So this is basically a phylogeny. It's called a phylogeny, and it's a reconstruction of evolutionary history. And usually what we do is we then map traits onto it. And so we expect certain things to come, certain traits to come before others. Why do whales have lungs and hip bones? Ancestry. They descended from animals that were on dry land, that used lungs, and walked and so had hip bones. So the hypothesis from Darwin, they have these characteristics, whales do, because they are descended from land mammals. In other words, mammals with 4 legs. If they did do that, we could predict if they had land mammal ancestors, then there would have been intermediate whale-like species with small legs. Test. You look for the fossils. Results. Darwin's hypothesis supported by indication of fossils with whale-like characteristics and legs too. Right? Here's the legs. Just starting to turn perhaps into flippers. So this animal probably lived in, shallow seas and things like that. Defining major geological time periods, we've had mass extinctions. That means where almost all the life on the planet disappeared, was wiped out. The two biggest ones, or the biggest one is the end Permian, where 90% of all marine species went extinct. The last one that was really big was the late Cretaceous, where an asteroid hit, and it looks like it wiped out the dinosaurs except for the birds. It wiped out about 80% of all taxa. So, again, in Permian. Now, there was also a late Cambrian extinction, an end Ordovician, and each of these extinctions, they're defined in age or periods. They're defined by the kinds of animals that you actually find in them. Now, this is all from about 500 million years ago, and there were a total of 5 really big mass extinctions of varying size, but also a whole bunch of little ones, because things change. But before 500 million years ago, there were also mass extinctions. There weren't hardly any multicellular organisms. It looks like at one point Earth was completely covered with ice all the way to the equator. But these are the ones we're going to concentrate on. And then, of course, that's the one that perhaps killed the dinosaurs. What mass extinctions actually do is they kind of reset the evolutionary clock. Some taxa make it and survive the extinction event, others don't. The ones that survive are the ancestors of future taxa that then evolve. Question is, do mass extinctions have major impact on the forms of biodiversity that exist later, or are they just kind of temporary reductions in biodiversity? Well, mass extinctions can have a major impact on biodiversity because only some taxa survive and others never to be seen again. Homology. Homology demonstrates that taxa, whether they're different species or higher taxa like birds and mammals and reptiles and things like that, they show that they had a common ancestor because they share structures, underlying similarity between structures. It could be anatomical, like these, in related species that result from common ancestry. So, these are all tetrapods, right? These are all the different kinds of bones. The humerus, the ulna, the radius, the carpals, the metacarpals, and the phalanges. Now, because all these animals shared a most recent common ancestor, they're all quadrupeds, 4 limbs, but they evolved over time different structures. But they still use the same bones. These are homologous bones. The same bones, that's what these the ancestor of all these animals had, first quadruped, and these bones have been used and shaped by natural selection. But they're all from a most recent common ancestor. So instead of creating new bones, let's say with birds, evolution can only work on stuff that is already there, existing things. This is called the panda principle. A panda has this thumb, but it's really not a thumb. It's part of it's actually part of, I think, one of the carpals. And it's used like a thumb, and it works good enough. So here we have the humerus, right? So that's the humerus in the seal. The bat also has a humerus. They're both mammals. A bird is not a mammal. They're more closely related to reptiles, other reptiles. Has a humerus. Another mammal. A horse has a humerus. We have a humerus. And a turtle has a humerus. If we go through this, we can actually see more things. Like, let's look at actually, we'll say the ulna. So it's this bone right there, and this is the radius. Well, the ulna is there, and the radius is on the other side. Here we can see it again. This is the ulna, and this is the radius. Carpals. Right here and right here. Oh, I didn't get all the ulnas, so ulna, radius, ulna, radius, and so on. So this is all underlying similarity between structures, in this case anatomical, and related species that result from a common ancestor. Same thing with the metacarpals. Okay. And the phalanges. They've all changed. In birds, the phalanges are hardly exist at all. It's the metacarpals that are making up most of the end of the wing. Bats is not the same. The phalanges, really, you can actually see it in a bat's wing. In the horse, the phalanges have been reduced and fused, so there's only one phalanges. Normally, with tetrapods, there's 5 different digits. Now, you can also have developmental homology. Here we have an embryo, 2 embryos. Okay. One's a chicken and one's a human. I think this is a human. You can see a bunch of things that as this as these embryos develop, they develop in the same way. Okay. They develop like here. Chickens don't have tails. Humans don't have tails. But we both shared a most recent common ancestor that had tails. Actually, the loss of tails for both taxa evolved independently, not at one time, because the ancestors of chickens were other reptiles with tails, let's say kinds of lizards or dinosaurs or whatever. And then the ancestors of humans were apes. They didn't have tails, but other monkeys that came before did. Another thing that shows up here is gills. Now, this is homologous because they both shared a most recent common ancestor back in time when the most recent common ancestor was a fish-like animal with gills. And so gills were lost only once, and then, but they show up as during development. So what's happening is during development, it kind of starts recapitulating or going over evolutionary events over time. So you have these earlier structures from ancestors that then later, let's say, disappear or change. So that's called genetic homology. No, I mean developmental homology, where, and the same thing here. This is of the circulatory system. This is what it looks like for a shark. And all these same things, and you can see the same thing in a human embryo. And then finally, we can have genetic homology. And we can say that these are different amino acids, or we could also say, let's say, they would normally, if they were genetic, they would be either a C or a T, or an A or G. But you can see, hold on a second. So this is a gene called aniridia. On the bottom, we have a fruit fly. And on the top, we have a human. You see how these are identical amino acids? Okay. Now, it turns out that fruit flies, we've learned a lot of stuff by crossbreeding them. And we've also favored certain mutations. So, a mutation here, this all has to do with genes for development of the eyes. This fruit fly is eyeless. And it went from a G to a D, and individual humans can also have had a mutation that makes them blind as well. So here's another one. Okay, so these are all changes. All these changes along the bottom here have to do with the fruit fly, and many of the same genes changed in individuals with aniridia, and they can't see. So this is all because of common ancestry. These alleles are from common ancestry. Vestigial traits. Structural relics that serve other functions in the evolutionary ancestors, but now have lost their functions or have new ones. Why do they demonstrate descent from a common ancestor? Okay, let's first take a look at the snake. Like the whale, snakes have lost their legs. In fact, actually, there's some lizards that look like snakes that have lost their legs too. So, but what you can actually see is you can actually see these bones, like the toes, phalanges, that you can see up here. You can see ankle bones up here. All these different things. Okay. So this is all blown up. And these are things that, of course, the snake no longer has, but they're actually vestigial bones that are no longer being used for walking around. Okay. Now, more vestigial traits. Goosebumps. No, I mean tailbone. Well, here's a human. We don't have tailbone anymore. We have a tailbone, but we don't have a tail. But we shared our most recent common ancestor, all the apes did, with a monkey that did have a tail. So monkeys came before, and then there was a split, and one lineage went off and became the apes, including the humans and hominins. Now, that meant there was this one change, like when I did that phylogeny, one change for losing a tail for primates, and that led to all those primates that became apes and humans, and the monkeys went off by themselves and still retain their tailbone. Okay. Now remember, evolution can only use what's already there. It's not like an engineer who can come along and design something brand new. Okay? And that's why there's a lot of things that are not really all that well designed. They're not designed. They're kind of a Mickey Mouse thing that selection managed to put together to make them somewhat useful. Selection, remember, didn't know this. There are just certain traits, and those that were favored increased and then changed over time. Okay, Now, there's also, and we can see how poorly certain things were designed or ended up that way, in a lot of, let's say, anatomical structures, and others as well. But one of them is the fact that humans are more likely to choke. And why is that? Well, unfortunately, okay, here we go. Unfortunately, we have the same opening right here that is used. So this is the oral cavity, right? What comes in here? Food and air. And here's the pharynx. And in humans, it's way up here. In other animals, it's way down farther. The epiglottis is the flap that results in a differentiation between the esophagus for digestion and the larynx and trachea for breathing. Well, that's a problem. First of all, it can get stuck. Let's see here. Okay. Now, it turns out that in our past, evolutionary past, humans and all other vertebrates, we had a common ancestor that was a fish that actually had a swim bladder and not a lung. Okay? And it was only because evolution uses what's there that as all these different changes came about, the lung actually eventually became, the swim bladder actually eventually became a lung. Like we have lungfish that are rudimentary lungs that don't work very well, but they work well enough. Well, we have a very good lung, and that's what we have. Unfortunately, all these lineages, reptiles, mammals, birds, all these fish, all had one opening. Now, normally, if I was going to design something like this, I would have two different openings, one dedicated to breathing, one dedicated to eating. But this is a holdover from our evolutionary past. So, because of this, we end up with a situation that's actually maladaptive. Now, other animals don't necessarily have a problem with choking, and that's because our larynx, I think, or the pharynx, has actually dropped in adult humans, but other animals have them in different places so it doesn't interrupt and cause problems in terms of being able to breathe or eat at different times. So because of our evolutionary history, we have choking. Humans are much more prone to choking, and frankly, choking is maladaptive. Choking kills a lot of people. You would think maybe selection would change that, but it can't because of all this, the way things actually develop now. Okay. Here we are. Goosebumps. Well, in our chimps and other great apes, they'd use the hair to stand on end for insulation, but also emotional display. We don't have hair anymore, except when we're still a fetus, but we still get goosebumps. Okay. And then, of course, we have the hair business. Now, we've lost hair, but it turns out once in a while, just so you know, once in a while, humans are born that actually have a tail. They usually get snipped off. They're not necessarily very functional, but they're definitely a tail. They can wiggle them. In the same way, some humans, because of the way genes are regulated, for hair, instead of the hair disappearing as an embryo, they retain it and they're born very, very hairy. In fact, it's called werewolf syndrome. So all these genes are still in place. This was all in the movie, but they're either dead genes, they've been shut off permanently, or they've been, with regulation, they're stopped from making their proteins. But sometimes you get a mutation and they turn them back on. So here we go, choking again. Why are humans prone to choking? It's our evolutionary history. In fish, the swim bladder is connected to the gastrointestinal tract. So here we are, actually. Okay. We have the swim bladder and the gastrointestinal tract. Okay. And different types of fish, the swim bladder can either be dorsal up here or ventral down there. In lungfish, the swim bladder is now, instead of being used as a swim bladder for buoyancy, it's now used actually to use oxygen from the air. So the lungfish has protolungs that are connected to the gastrointestinal tract through a ventral T-junction. And that's right here. Okay. Now in reptiles, in fact all tetrapods, the airways and the gastrointestinal tract cross at an X function, X junction. Okay. So here we are. We've got, I think this is the, let's see. Okay. Okay, so we have the gastrointestinal tract and the airway is red and blue. Same thing here. Okay, and then here's different things. Now, it's thought that there's a newborn infant, and things are different between an adult and a newborn. It turns out the pharynx for adults actually drops. Newborns don't have this problem. These are Neanderthals. I think they probably spoke as well, but like other vertebrates don't have this problem. Tetrapod vertebrates don't have this problem because the structures of all these different things where they cross at different places. And so they're much less likely to choke on food. But this is a real problem. This is also maladaptive. You might wonder, why doesn't evolution by natural selection get rid or change this so it works better? I mean, you would never design a system like this, but evolution is often constrained from prior evolutionary, how things evolved previously. And this is one of those cases. Another example, now these are genetic and molecular. Humans have 23 pairs of, or 23 chromosomes, 2 pairs of each, for a total of 46. So 23 from mama and 23 from papa. They have the same genes, they're homologous chromosomes. It turns out that all the other apes have 24. Okay, so right it here, the initial, these are the oldest, like the orangutan. They shared a most recent common ancestor with humans about 10 million years ago. So in the beginning, they're apes, 24 chromosomes. This group, still 24 chromosomes. Now this group, this is where somehow we only have 23, while our chimpanzees or bonobos still have 24. The question is, how did that happen? Well, it turns out that if you were just to drop one chromosome, you would end up with an organism that wouldn't develop at all. It'd be dead. Viability selection. Okay, so, but what actually happened is that human chromosome 2 is actually a combination of ape chromosomes 12 and 13. And we know this because chromosomes have what are called centromeres, okay, where the long arm and the short arm are joined. And also where the other chromosome, the homologous chromosome, is also joined as well. Okay? Now the thing is, what we found in humans is we've found centromeres of human chromosome 2 is actually the telomeres, these are the ends of the chromosomes, of ape 12 and 13. So what happened was, the most likely and most simple explanation is that these 2 chromosomes on the apes, in terms of what our human ancestor, they actually fused at some point into one chromosome, which is human chromosome 2. Now, that's very strong evidence for us and great apes actually sharing a most recent common ancestor. Okay. So here's a phylogenetic tree again. I didn't really go over this quite properly, but wherever you either have branches and then you have nodes. So right here is a node where one group, lineage, and another shared a most recent common ancestor. Okay? Now, it's incorrect to say that monkeys and humans, or humans evolved from monkeys. About 28 million years ago, we shared a most recent common ancestor with this branch of monkeys that includes macaques, squirrel monkeys, baboons, all kinds of different things. And then this other branch right here resulted in the apes. Okay, so we have the family Hominae, and then we have the subfamily Hominiminae that doesn't include the orangutan. And then, of course, we have a much more recent common ancestor where there's the homonyms, that's us, and then chimpanzees and bonobos. Okay, so we share the most recent common ancestor with monkeys, as well as all these other things like orangutans, gorillas, common chimpanzee, bonobo, and us all share the most recent common ancestor. So at one point there was right here, this lineage, there was a change in that they all lost a tail. A tail was lost. And so they all inherited not having a tail. Okay? Whereas these monkeys actually had a tail. They also had a bunch of other things like increased flexibility of the thumb. Many different things. Delayed sexual maturity. And then around 7 million years ago, the clade, these are nodes, and these are branches, of course. And a node is where there was one lineage split into two. So here's a lineage splitting into two. One went towards the chimpanzee and the bonobo, which then subsequently split about 1 million years ago. We split from this lineage about 7 million years ago in terms of our human ancestors. Differences between homology and analogy. So you can also have a situation where it looks like you have two species that are similar from descent, but they're not. For example, these two bird species, they're actually on completely different branches of these trees. But in the environments that they're in, long tails are favored. Okay. Now, homology. Remember, homology is all about descending from a common ancestor, and so you have homologous structures like anatomy, let's say the bones that we all see in vertebrates, or development, the same sort of development in related taxa, vestigial traits. Those are all examples of homology, and including genes that we share from, let's say, very long time ago that are homologous alleles. Okay, now analogy is similar traits, we'll say like this tail, long tails, that might seem like they're in fact related by descent, but they're really not. They evolved independently twice. Here we have these long legs. Well, it turns out these are homologous traits, the long legs. There was only one change, a split from this other taxa with short legs, and the common ancestor actually had long legs, and so both inherited long legs. Now, we actually have to know whether something is analogous, in other words, not related by common descent, and which stuff is by common descent when reading an evolutionary tree. So when you have traits that are similar not due to ancestral descent, but perhaps due to similar environments, then you have these. If you try to do a phylogenetic tree on that one trait, like these tails, you would be led astray. It's actually called homoplasia. You don't have to know what that is. Okay. Now, here's an example of homology, or not homology. These two, this is an ichthyosaur, and this is a common dolphin. An ichthyosaur is actually a kind of reptile. A dolphin is a mammal. they look similar because they live in the same environment. Their shape is similar because it's easier to move through the water with that kind of fusiform body than it is with things all spread out. But these are genes right along here. These are developmental genes called Hox genes. And it turns out that all animals share Hox genes. Now, we share these genes. Let's see, move it up. Okay, so humans, fruit fly, and humans. They share all these similar Hox genes, but mammals have a few extra others that have been added on. New genes that have evolved for development. But they share those same genes because of common ancestors. So again, up above, even though it looks like very similar creatures, one's a mammal, one's a lineage of reptiles that went back to the ocean and lost all the legs. They may share the same trait again. They look the same because they lived in the ocean. They had to travel through water. But those features are not because of descent from a common ancestor. They're evolved independently. Okay? And then the Hox genes, on the other hand, we share a lot of the same Hox genes with fruit flies. And these are a whole bunch of different ones, right? So, and you can see that, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. I guess fruit flies have up to 10. I think we have up to 13. So eventually, in fact, even before the fruit fly, there were fewer Hox genes. And then there were mutations, and new Hox complex genes were added on, or they evolved, and they took on new functions in terms of how organisms develop. So we actually share those because of a common ancestor. Okay, ancestor-descendant relationships. Okay, so like I mentioned, this is a phylogeny. Old, young. So time goes this way. Nodes, of course, are right where two branches come together, and then we have branches. And then what we've also done is we've mapped character states onto this phylogeny about when the characters actually showed up. Okay, so we start out with a lungfish. Now, before the lungfish, there was a fish. At a certain point, lungs evolved, rudimentary lungs. And so we ended up having a clade with lungs, including all of these. Now, the same thing happened in terms of limbs. As we saw with those transitional fossils, you had fish that had these, their fins evolved differently so that they could actually be used to semi-walk around in a kind of a watery, shallow watery environment. Okay, so we've got limbs happening here, 4 of them, and all of these things actually share limbs. And when that happened was, I was during the Devonian period. Now, frogs and salamanders do not have an amniotic egg. But all the other ones, all the different reptiles and mammals and birds and dinosaurs, which were all extinct, They all have an amniotic egg, which protects the egg from desiccation, from lack of water. So frogs and salamanders are only found, they have to be near water, and if they're not near water, they will dry out and die. All these other guys, they can be near water, but they can also be away from water and still survive. Okay, so mammals, fur and lactation. This hole in the skull below the eye socket happens right there. We don't have that. Mammals don't have that, but all these other ones do. For turtles and tortoises, all the bones were fused to form this carapace. That's the only lineage that has it. Extensive change in skeleton. Here, for lizards and snakes, determinate growth, they shed their skin. Those are the things that lizards and snakes have that none of the others have. Here, hole in skull in front of the eye socket. So, crocodiles and birds. Crocodiles also have, by the way, a four-chambered heart. Birds also have a four-chambered heart. And they have that because their common ancestor had a four-chambered heart. Not shown here are dinosaurs. So birds are actually one branch of dinosaurs. And of course, birds and dinosaurs had a number of things in common, especially they had some bone structure in common, but also feathers. Okay, so we've mapped these states out onto these different lineages, and remember we're going from old to younger, so this is how these first limbs for tetrapods came along, then amniotic egg, then hole in the eye socket, determinate growth, four-chambered heart came much later. Okay, now mammals also have a four-chambered heart, but not because of common ancestry. It's actually because it evolved separately and independently. And the lungs that we share, that we have, are actually very different from the lungs that you find in crocodiles and birds. So phylogenetics, the study of evolutionary relationships, ancestor-descendant relationships. How might we represent these relationships? Well, this is called a phylogenetic tree. And so we can see whole groups that share a bunch of things in common because they have similar traits. And other groups that share things in common but not with other previous groups. How do we tell if a species is related? Well, we can use physical features. Let's say a vertebral column. That's common in all vertebrates. All these are also quadrupeds. They have four limbs. But there's a turtle, there's a beaver, so here's a lineage of reptiles. Here's a mammal, a beaver. These are two mammals also. One is a marsupial and one lays eggs. But beyond that, here's this node. This is called a polytomy instead of just two lineages splitting off. With this phylogenetic tree, we really can't tell much. We have to know if there's differences, characters that certain groups have and others don't. So we know the turtle doesn't have hair and mammary glands. Well, we know one thing. If they're all mammals, they have hair and mammary glands. So we can say that we don't know how these are related. They all have the same place where they share a common ancestor, but we do know that at one point there was a mutation that resulted, or a couple of mutations, one that resulted in making milk and the other was starting to make hair. Okay. Now, with this phylogenetic tree, there's traits that actually separate a platypus from other mammals, marsupials and placental. Okay? Well, it turns out all these things that were previous, they all laid eggs for the most part. Okay? And the platypus also is a monotreme, and it also lays eggs. It's one of the earliest mammals that you would find, and there were probably, in the beginning, before there were anything else, there were mammals that laid eggs. Okay? So, this right here is when you actually had gestation, where the mother, the egg was retained in the mother, the embryo grew. Then marsupials, they have a little pouch and the embryo comes out at a very, very, very early in development, crawls up and into the pouch and attaches to a tit. Okay, and then eventually we ended up with placental gestation, which evolved independently, and a beaver, and a whole bunch of all the other mammals that were placental, elephants, everything like that. Okay, so this is how we know we map these traits, and we can actually then, if we have traits that are different from one group to the next, we can then map them in perhaps the order that they actually arose. So here's a phylogeny of actually a lot of reptiles. We actually have some outgroups. Rana is actually not a reptile, an amphibian, it's a frog. We have a bunch of reptiles. We have crocodilians way down here. And then we start getting into a group of dinosaurs. And there's two groups of dinosaurs, this group, and then this group. Okay? And then they diverged again into separate lineages at various time periods, and you can see the time periods down here about when it happened. Now, all of these from around here are called tetrapod dinosaurs for the most part. They stood on two legs and had smaller forelimbs. Tyrannosaurus rex, Oviraptor, that you might remember from Jurassic Park. And then right around here, aves, this is right here, this is where the birds split off from a common ancestor, Deinonychus. They shared many similar characters. They were dinosaurs, they had feathers, all that, but they didn't have flight feathers, and they were far too big and heavy to actually fly. Okay? And then from then on, we have this radiation of birds. And here's a bunch of all the various lineages of birds, like corvus, which is the corvids are jays and ravens, magpies, crows, things like that. Okay? And then, let's see. What is that? Oh. This, now, remember this is actually all mammals. But you can see that this group forms its own clade, and it actually shared a most recent common ancestor way back when. Okay? So that's a dinosaur phylogeny. Okay. Traits are nested in a hierarchical pattern because of time. When a trait that you see in organisms, they show up at different times in the record, in the fossil record. They show up, those that show up early, all descendants from then on probably have at least a version of it. Those that show up later, only some lineages have it, others don't. And so here's the fossil record. This is old and younger up here. And you can see where we start to find fossils. And we found fossils with birds much later than the first reptiles, but we see that they're similar. The same thing with mammals. Okay. Now remember, if we're looking at a phylogeny, it goes from old to new, or more recent. So mammals and birds are more recent than when amphibians first showed up. Okay, now that means what we mean by nested, for example, in this phylogeny, is that all these individuals have a backbone, the vertebral column. But the fish doesn't have 4 limbs. All of these have 4 limbs. Later on, the amniotic egg was formed or evolved, right? And so we have these, including reptiles. So all of these things fit inside of the other. Everything, this whole branch here with everything fits inside of the fish because they all have a vertebral column. Okay, now this is all because of time. Here we have the oldest, the Ordovician, and then up here is recent. Okay, now the first jawless fish in the fossil record is right down here between the Ordovician and the Silurian. Okay, so here we are. Okay, first jawless fishes from an ancestor that maybe didn't have a vertebral column. Then there was this group of fish that were called placoderms. We don't know exactly when they split off, but at a certain point, namely the Permian, they all went extinct. So we don't have any placoderms today. Later on, around the Devonian, we start to see cartilaginous fish like sharks and rays. They don't really have bones, they have cartilage. Okay. And perhaps in the same, we also see bony fish, but we actually assume, in fact, that bony fish came after the cartilaginous fishes. And then sometime in the Devonian, we ended up with tetrapods. And then reptiles, these are all amniotes. Okay. And then earlier reptiles, mammals split off. And then later, the birds split off from another group of reptiles, younger reptiles, and it wasn't just the birds, it was the dinosaurs. And eventually, the only ones that were left were, in fact, so these are all dinosaurs, okay? And the dinosaurs went extinct when the asteroid hit, and the only dinosaurs left over now are birds. Okay, so mammals and birds actually, they split off at different times. Okay, now, so you can see anatomy of modern forms suggests an order in the evolution based on time. In terms of the fossil record and also genetics, we know that in fact the first thing that actually, the first tetrapods were actually amphibians. They weren't mammals. They were amphibians. They were the lineage that led to amphibians, reptiles, and mammals. Okay, and the reptiles, of course, included a bunch of things. They included lizards and snakes. They included turtles and tortoises. They included crocodilians and birds and dinosaurs. Okay, and this is all in the fossil record. So we would never expect to find, let's say, a bunny rabbit way back in the Devonian. Okay, there's an order, and this order is based on time. So a nested pattern means certain traits found within that that only a certain group have, compared to relatives that have only that they all have. Okay, so let's look at this nesting pattern. Okay, tetrapods are the oldest. They have 4 legs. Okay, all of these have 4 legs. Amniotes, the ones that have a covering for their eggs so they won't dry out. These all have amniotes, all of these, including birds, reptiles, and mammals. And then, of course, within, only the birds have feathers. Okay? So you can see there's a pattern because over time, things evolved. First, there was limbs. Then there was an amniotic sac. Then there was a number of different things, including, depending on which lineage of reptiles. And then finally, there was feathers. And feathers didn't evolve in birds, they actually evolved in dinosaurs that preceded the birds. So I want you guys to get this idea that there's this hierarchical pattern. The earlier a trait shows up, all the descendants of that ancestor have that trait. A more recent ancestor will have a trait that none of the previous ones actually has. Okay? So the traits are actually nested one within the other based on time. In this case, the outer layer is the oldest, and then the youngest layer are the birds. So this is the tree of life. There's 3 domains: bacteria, archaea, and eukarya. We're all eukaryotes. In other words, we have cells with a nucleus and organelles like mitochondria. Okay. Um, there are actually, in life, and in all living things, there are three great domains. There are the bacteria. There are archaea, and then the eukaryotes that have a nucleus and organelles. And some of these are plants, some of these are animals, some of these are ciliates, all kinds of different things. Now, it turns out that archaea and bacteria, even though they don't have nucleuses, they're not as closely related as archaea is more closely related to eukarya, because we share a node, than we eukaryotes are to bacteria, or archaea are to bacteria. Okay? So, here we have all of life. Now, this is all based on rRNA, which is it's ribosomal RNA. It's showed up. All living things actually have ribosomal RNA. This is where things get turned. Instructions get turned into proteins. Okay. Now we know that all life also contains a DNA. Actually, not quite. I consider viruses living things, even though they can only live with other things that actually have cells and metabolism. Okay, so there's some viruses that only have RNA, but they do have amino acids and lipids and all that kind of stuff. And then all life contains semi-conservative replication, mutation, and DNA repair. And then also the transcription from DNA to RNA and translation from RNA into various proteins and amino acids and then proteins. And then we share a lot of metabolic pathways. So this is how we know that, in fact, all life right now shared a most recent common ancestor, and perhaps 3.5 billion years ago. It's hard to know.